Hi, everyone. Good morning. I am excited to start our final day of the conference. I hope you've all enjoyed all of the information. I know I have. This morning, we have the pleasure of bringing Dr. Peter McGuire to you to talk to you about viral infection and mitochondrial disease. Dr. Peter McGuire received his MBBCH with honors from the Royal College of Surgeons in Ireland in 2003. Following a combined residency in pediatrics and metabolic genetics at Mount Sinai Medical Center in New York City, he remained as an assistant professor in the program for inherited metabolic diseases at Mount Sinai. Dr. McGuire is board certified in pediatrics, clinical genetics, and biomechanical genetics. In 2010, Dr. McGuire moved to the National Human Genome Research Institute, the NHGRI, at the National Institute of Health to join the Physician Scientist Development Program. He was appointed to the position of tenure track investigator in 2016. Throughout his career, Dr. McGuire has been focused on improving the care of patients with disorders of mitochondrial metabolism. By combining his training in immunology and biomechanical genetics, he has fashioned a translational research program to understand the interplay between mitochondrial metabolism and the immune system. As head of the Metabolism, Infection, and Immunity section, or MINIS, at the NHGRI, Dr. McGuire and his team study the interplay between metabolism and the immune system in patients with inborn errors of mitochondrial metabolism. The group focuses on two aspects of immunometabolism. One, the immune system activation and end organ mitochondrial metabolism. The focus of the group's research on immune system activation and end organ metabolism is based on the clinical observation that infection is a major cause of morbidity and mortality in patients with mitochondrial disease. The MINIS uses animal models combined with infectious organisms to yield insights into the metabolic seen in disorders of mechanical metabolism during infection and to identify potential targets for intervention. The second area of study is the role of mitochondrial in immune cell function. The group also studies mitochondrial metabolism and immune cell function. Immune cells drastically alter their metabolic programming during activation and differentiation. The deficiencies present in patients with mitochondrial disease may affect these processes. The group developed a clinical protocol in the NIH Clinical Center called the NIH Mini Study. You can find information about that at the clinical trials, clinicaltrials.gov. Immune phenotypes identified in patients are further explored via animal and cell culture model systems. By expanding the immune phenotype of patients with mitochondrial disease, these studies will have an impact on the clinical care of patients as well as serving as the foundation for understanding the role of mitochondria in immune function. Whew, that was a lot, Dr. McGuire, but I'm so excited to kick this off and take it away.
Sure, everyone can see my slides. Is that correct? Okay. So first of all, I would like to thank the organizers for the opportunity to speak with you today. And I'm going to talk about viral infection in children with mitochondrial disease. Now, realizing that this is a conference that is for patients with fatty acid oxidation disorders, I like to think of the two disorders as like cousins. So a lot of things that can happen in mitochondrial disease, of course, can also happen in fatty acid oxidation disorders. And mitochondrial dysfunction can be a part of fatty acid oxidation disorders. So the mission of our section at NIH is to define some of the risk factors and mechanisms that are involved in infection-related clinical decline in children with mitochondrial disease. And to do that, we start with clinical research. So we have patients who come from all over the country to the NIH Clinical Center. And from that, we form questions about their disease and why they may be having difficulty with infection. And then we develop model systems. And those model systems may be based on animals or may be based on material that we collect from patients. And the ultimate goal is to understand why Some patients with mitochondrial disease may have difficulties with infection and may decline with infection, and then there are some that actually don't. So what are the differences between those individuals? And by understanding that, can we eventually develop treatments which will be able to help these patients? So this is our clinical study, the NIH mini study, which stands for Metabolism, Infection, and Immunity. And as Stephanie mentioned, you can find the listing on clinicaltrials.gov, and that's the number, the clinical trial number up there. And this is really a natural history study. So basically a study that follows patients in time to understand infection and immunity in children with disorders of mitochondrial metabolism. So what we do is we look at things like patient's infection history. We look at their immune function to see if that is able to fight off different infections. And then we also look at the disability that may actually develop as a result of having an infection. So I know everyone's intimately familiar with COVID-19, and definitely worth, in a talk that is about viral infections and mitochondrial disease, it obviously is worth mentioning. So this is Johns Hopkins University's COVID-19 dashboard. It's a really great resource for following the trajectory of the pandemic. And you can see here, this is from this past week, where there are 34 million cases in the US and over 600,000 deaths. The vaccines, which are currently approved for children 12 and up, or at least one of them is approved for children 12 and up, there have been 337 million doses that have been administered, but this only constitutes roughly 49% of the country. So there's still a lot more work to do in terms of getting people out there vaccinated. So what have we done to address the COVID-19 pandemic? So we have the mini study, which is represented here by the orange arrow, which is an ongoing study. And through the mini study, we initiated a number of other studies to try and understand the impact of the COVID-19 pandemic on children with mitochondrial disease. So we started by giving a questionnaire online to the mitochondrial disease community about the COVID-19 pandemic, which you can see down here. And I'll tell you about some of the data that we collected with regards to that. Then also we started protocols looking at COVID-19 infection, where families contact us if they have a child who has been affected by COVID-19. We also initiated a study, which is a home study, which is still ongoing, about viral exposure in children with mitochondrial disease. So this looks at what different viruses they may have been exposed to. And then finally, vaccination in COVID-19. And this is a study that just began recently with beginning to collect some information on people's experience with the COVID-19 vaccine itself. So to jump right into it, so what are viruses? So viruses are basically submicroscopic infectious agents, so very, very tiny. And the interesting thing about viruses is they infect all different forms of life, from plants to insects to animals to humans, as we know. And you can see here on the left, they come in all different types of shapes. So you can see here herpes viridae, which is the family of herpes viruses, which cause cold sores, which a lot of people experience on the lip, or orthomyxoviridae, which is your influenza virus, which is around every year. And the thing about these viruses is that they are not necessarily considered living things. In fact, they actually need living cells to actually replicate. So what happens, which I'll show you, is that viruses take over the machinery of our cells to be able to copy themselves and make more viruses. So having a living cell is required. The core material of viruses is either DNA or RNA, which leads scientists to kind of divide these into either DNA viruses or RNA viruses. Okay, so these are some of the shapes which I showed you here on the left. And what I'm showing you is some of the parts of a virus. So this is just kind of a schematized version of a virus where you have essentially the viral envelope. Some viruses don't have envelopes. but this is showing an envelope virus. So like the coronavirus is an envelope virus, and on the outside are these envelope proteins. And the one we've been hearing about all this time in terms of coronavirus is the spike protein, right, which is important for coronaviruses to be able to infect cells. Now, inside, there is the viral genome. And as I mentioned, that's either DNA or RNA. And that's covered by a nucleocapsid. So in other words, a protein structure that helps protect the DNA or RNA within the virus. So unfortunately, viral infection is inevitable in children. It's somewhat unavoidable. So this was a study that was done back in the 2000s. This is the Copenhagen Prospective Study on Asthma in Childhood, or COPSAC 2000. And this is a prospective cohort. So what that means is they take kids and they follow them from birth up until 3 years of age. And they look at the development of asthma, but most importantly, which is interesting for us, is that they look at essentially infections and when they occur over this 3-year period. So there are about 411 individuals. And what you can see is that most children between the ages of from birth to 3 years of age have between 10 to 15 infections over that time. And the majority of those infections are actually respiratory tract infections. So coughs, colds, runny noses, things like that. And also what you can see here, which I'm sure a lot of patients and families and caregivers are intimately familiar with, is a lot of these things occur over the winter season. So there is a seasonality to respiratory viral infections. So what is the seasonality of respiratory viral infections? Well, here there are a number of different viruses listed on this table, and it goes through the different months of the year. So you have your winter viruses, which we're familiar with, such as influenza virus. This is a coronavirus, but this is a coronavirus that causes cold-like symptoms, not the pandemic coronavirus, which we're experiencing now. And then respiratory syncytial virus, that's another one. Bocaviruses and adenoviruses are respiratory viruses that occur all year round. And then there are parainfluenza viruses that have certain times that occur during the year, as well as human metapneumovirus, which is throughout the year, and rhinovirus, which is throughout the year. And then there are summer viruses, such as the enteroviruses, which a lot of us experience sometimes during spring, but mostly into the summer. So how are these viruses actually transmitted? So there are a number of ways. So you can see here on the right, one way is by touch. So in the beginning of the pandemic, there was a lot about hand washing with SARS-CoV-2. As time went on, they realized that that type of transmission, although it does occur, is the risk is actually much less than respiratory spread. Influenza as well, by sharing things like, not only touching surfaces, but sharing utensils or sharing other things between individuals can lead to the passage of these viruses. Respiratory droplets, once again, SARS-CoV-2 and influenza. By direct contact, so things like Epstein-Barr virus, which causes mononucleosis, used to be called the kissing disease, and then human papillomavirus. And then also there are bloodborne transmissions, such as HIV, hepatitis C, and Ebola. Contaminated food or water, so noroviruses. So anyone who wishes to come in contact with a norovirus, you can try taking a cruise, because those are the viruses, or one type of virus at least, that causes outbreaks of illness on cruises. Insects, the Zika virus, which you've heard of probably from a couple of years ago, which causes birth defects in individuals, a small head essentially, or microcephaly, that can be transmitted by mosquitoes. And then during childbirth, things like cytomegalovirus can also be passed, which can cause birth defects as well. So what are the factors that affect virus transmission? Well, there's a lot of different factors. Basically, this is an interaction between the host and between the infectious agent, such as the virus. So there are seasonal factors, such as temperature, humidity, sunlight, as well as vitamin D status. Vitamin D is very important for the immune system and health of the immune system. And a lot of individuals are vitamin D deficient because we just don't get enough sunlight out there. And the availability of vitamin D in various foods and stuff is relatively low. Indoor temperature, things like temperature and humidity, or the air change rate, whether it's recirculated air, especially for droplet-based infections like influenza or SARS-CoV-2, host defense properties, such as how good is your antiviral immune defense? How good is your immune system at fighting off those viruses? Replication between viruses, and an efficiency of transmission, or the ability of viruses to enter your respiratory passages. And then also the properties of the virus itself. How viable is the virus outside of its host normally? What are the droplet dynamics? Do they spread very easily? How big are they? How small are they? And then also issues of between individuals, airborne or aerosolization of virus, droplet spray or direct contact, and then indirect, such as fomites. So fomites just small, things like dust and small particles that viruses can stick to and serve as kind of a way of transmitting between individuals. And then also human behavior. So this is something that we've become intimately familiar with during the pandemic, where interactions between individuals were, for a period of time discouraged, and social distancing was encouraged, et cetera. So there's a lot of aspects of human behavior which we've learned about as a result of the current COVID-19 pandemic. So what are some of the symptoms of viral infection? So this is a talk all about COVID-19, but the nice part of COVID-19 as an example is that it has many of the symptoms that individuals can experience with a viral infection. So you can have things like cough, shortness of breath or difficulty breathing. You can have fever and chills. You get a muscle and body aches, and you can have vomiting and diarrhea. And loss of taste or smell is something that is somewhat unique to SARS-CoV-2, although with other viral infections, when you have nasal stuffiness and things like that, your sense of taste can change. It's just that it may be longer lasting in SARS-CoV-2. So depending on what type of virus you may actually get, these symptoms can change a little bit. Not all of them have respiratory symptoms, so it really depends on the type of virus. Okay, so how do viruses infect cells? So as I mentioned, viruses are not necessarily considered to be living. They actually require a host cell to be able to replicate. So here we are looking at, just as an example, SARS-CoV-2, but this whole pathway that I'm showing you, which I'll walk you through, really shows how a virus kind of leverages the machinery inside cells to replicate itself. So this would also be very similar for things like influenza and other respiratory viral infections. So first of all, the virus has to bind to a receptor on the surface, and then after it binds to a receptor on the surface, it gets internalized into the cell. And then once it's internalized, it breaks open, and the genetic material inside, in this case it's RNA, is released. And once that's released, the cell, so these things are called ribosomes, that actually takes the RNA and translates it and makes proteins out of it. And these proteins are pictured down here. And these proteins are important because it helps you reassemble more viruses, essentially. So then also the host genome, or the RNA or DNA, in this case RNA, has to be replicated. And then all of this is repackaged again into these viral particles, which then are released from the cell. So even though, as I said, this is kind of a coronavirus-based drawing here, this is applicable to a lot of different viruses and how they infect cells and how they're repackaged. So how do we protect ourselves from viruses? Well, but it's by the immune system. So the immune system is very important because it helps protect us against all different types of infection, whether it's bacterial infection or parasites or fungi, even cancer and pollution. But today we're going to talk about viruses specifically. So the immune system has many different parts to it. You have barriers, such as things like mucous membranes and the skin. And then you have immune organs, such as the tonsils. The tonsils are important immune organs in the back of your mouth and your throat. Lymphatic vessels, the thymus, which is important for making immune cells. The lymph nodes, which are all over the body, but these are showing some in the armpits, as well as down here in the groin area. There are also lymphatic vessels, which Really, this is kind of underplaying what lymphatic vessels look like. They're actually a highway system that stretches all over the body, which connects different parts of the immune system and serves as a highway system for immune cells to kind of traffic throughout the body. Then, of course, there's the bone marrow, and the bone marrow is where the immune cells are actually made before they leave and go to all these different immune organs. So how does the immune system protect us? Well, the immune system has to essentially interact with infectious organisms, and that can be done by either natural infection or that can be done by vaccination. And in this way, the immune system learns about that organism and basically how to fight that organism. So there are essentially two arms of the immune system. So there's what's called the innate arm and the adaptive arm. So the innate arm, which you can see down here on the left, is the rapid response, right? This is what kicks in right away. It involves a number of different cells like macrophages and neutrophils and things like that. And you can see here on the right, we have a timeline of the immune responses and how the innate and adaptive immune response relate to each other. So when you have the entry of a microorganism, that microorganism, like a virus, basically can replicate, and it's replicating within cells. And this is the establishment of infection. And during that time, the innate immune response becomes activated and tries to prevent that from happening, or at least hold it off until the adaptive immune system can actually take over. And the adaptive immune system includes things like B cells, which make antibodies, and T cells, which kill virally infected cells. And while the innate immune system is kind of holding off the infection, basically what you have then is the kick-in of the adaptive immune response. So the T cells and B cells, which basically then serve to decrease the amount of virus in the body until the pathogen or virus is completely cleared. And then the thing that's important is after doing so, these B cells and T cells can enter a memory phase. So in other words, this is how they learn. They've seen this before. So the next time that you actually encounter the virus, you actually have a more robust response. And that's what I'm showing right here. So these different circles here are different immune cells. So these are T cells and B cells, and the Y-shaped things are antibodies, which are made by the B cells. So when you have the primary immune response, which I just kind of showed you, where you encounter essentially the virus, you have an expansion of T cells and B cells. They get rid of the virus, and they learn essentially about that virus. So this can persist, this immune memory or this learning can persist for months and years. And then when they're exposed to the virus again, they have a secondary immune response where you get more T cells and more B cells and more antibodies. And individuals either experience a very attenuated version of a viral infection, or they don't experience any viral infection at all. And this can happen, and this works not only for infections, but it also works for vaccination. Vaccination works exactly the same way, where in this early period here, you're introducing the vaccine, and then later on, if you actually, so for instance, for measles, if you actually encounter measles and you've been vaccinated, you'll have a very robust response and you won't experience measles. Okay, so how does the immune response actually work? How are these T cells and B cells actually getting rid of virus? So the first thing is T cells. So the T cell here that's really important is something called the CD8 T cell. And what it does is it attaches itself to target cells. So this target cell is a cell that's infected by a virus. And what it does then is it dumps a whole bunch of stuff you can see here in this cartoon, which basically then kills this virally infected cell. So T cells are important for killing virally infected cells by essentially dumping a whole bunch of immunoreactive substances onto that cell, where the cell will essentially shrivel away and die. Then there's antibodies, right? So antibodies are made by B cells. And this is a very complicated cartoon, but I'm basically just walk you through it and give you the highlights. So this is what we showed before, where a virus essentially is entering a cell, right? And the virus needs essentially the cell to replicate itself and make new viral particles, which can then escape. Well, antibodies can help block this process. So for instance, you see antibodies here binding to the virus itself. You can see antibodies preventing the virus from exiting from the cell. So antibodies are really important for essentially blocking the virus, either blocking the virus from interacting with the host cell that it can replicate, or binding up the virus so it can actually be eaten by the immune system. Okay, so, understanding this about, immunology and how the immune system actually works, can we use this knowledge in any way to understand what's going on in individuals or children with mitochondrial disease? So can we look at essentially this production of antibodies against various viruses? So I'm going to show you some data. This is from a study that we did using a platform called VirScan. And what VirScan is it's a way of fishing out antibodies from the blood against different viruses. So you can see here, this is essentially something called a bacteriophage, which is a virus itself, which you can put different parts of a virus inside, and then it will express that on the surface. And so this is like a library, essentially, of all these different viral proteins, which represent hundreds and hundreds of different viruses. And then we can fish out the antibodies in an individual from their blood and see essentially what viruses were they actually exposed to. So this is a really powerful, powerful technology. So first of all, number of viral exposures. So when we looked at roughly 15 patients with mitochondrial disease and about 13 patients who were age and sex match controls, we found that, so these are individuals who are less than 10 years of age. The average age is about 6 or so. And you find that those individuals were exposed to roughly 15 or so different types of viruses, right, over their lifetime. So this is a snapshot. This is a snapshot at about roughly 6 years of age. They've been exposed to about 15 different viruses. You can see there are some individuals with mitochondrial disease that have higher levels, where it's about 20 or so. But in general, there was no difference between the groups. As we collect more samples and more families, looking at this, these numbers may change. But so far, it looks like roughly about 15 different viruses they are exposed to. Okay, so this is the rogues gallery. So who are the culprits here? So the interesting thing is that it's very similar between individuals who don't have mitochondrial disease and those who do. And some of the top hits, so the percent positive, the number of individuals that we looked at who were positive for that virus. You have rotavirus, A and B, that causes things like diarrhea. You have human beta herpes virus. So this is something that causes roseola, or something that's called slap cheek, where kids get a very rosy type of infection on their cheeks. There's enteroviruses, there's respiratory viruses such as RSV and human orthopneumovirus, as well as rhinovirus and other enteroviruses. So this essentially is the top 10 list of the viruses that individuals with mitochondrial disease may be exposed to. And this is important to understand, which are the viruses, and therefore, are there any strategies that we can eventually develop that can help us with these different viruses. And so when you look at these different viruses, this is a longer list. So the top 10 is up here, but this is a much longer list. We asked the question, are there any viruses that individuals with mitochondrial disease seem to be exposed to or have antibodies against, whereas where they don't appear in control individuals? So positive in mitochondrial disease, negative in control individuals. And if so, what can we learn about that? Well, what was interesting is if you look here down at the bottom, human alpha herpes virus, or herpes simplex virus, right? This is the virus that causes cold sores in the mouth. And it's rare to have antibodies against HSV1 or herpes simplex virus 1 if you're less than 10 years of age. That's kind of rare. And you can see here in our controls across the bottom, green, the more green it is, the more positive it is. These are statistical tests. But all you have to take away from this is the more green it is, the more positive it is. And you can see here in the controls, it's all white. So essentially, they don't have any exposure to or any evidence of exposure to herpes simplex virus by looking at antibodies. Whereas you can see in mitochondrial disease, or 15 patients, about 1/3 of them, or 5 of them, have antibodies against herpes simplex virus. So that's kind of strange, right? Because most kids under 10 don't show exposure And this is based on a large database that we looked at of thousands of children who don't have, where essentially the levels of antibodies to herpes simplex virus is very low. So in general, kids less than 10 don't have it, but our mitochondrial disease kids, 1/3 of them seem to. So, what is it about herpes simplex virus and mitochondrial disease? Well, it turns out that HSV1 infection, or herpes simplex 1 infection, does something to mitochondria when it actually infects cells, right? So when herpes simplex virus enters cells, it actually depletes mitochondrial DNA. So, and that's by a protein called UL12.5. And this is what I'm showing you here on the right. This is essentially these are cells that are infected with or have this protein from herpes simplex virus, UL12.5, put inside of it. And this is the mitochondrial DNA, which I'm showing you here in the control or the parent. And this is the nuclear DNA. And when we put in this UL12.5 protein, what happens is the mitochondrial DNA disappears. So herpes simplex virus, when it infects cells, chews up mitochondrial DNA and gets rid of it, which I'm showing you here on the right. It's very low. So what does this mean? Well, when you deplete mitochondrial DNA, you inhibit the mitochondria and mitochondrial function, oxidative phosphorylation. So therefore, herpes simplex virus seems to require an inhibition of oxidative phosphorylation to be able to replicate and form more HSV particles. Patients with mitochondrial disease already have depressed oxidative phosphorylation, right? Their mitochondria don't work very well. So therefore, are they more susceptible to HSV infection, or are they more of a natural host for HSV1 infection? And this is something that we're currently looking at. Okay, so what are some of the risk factors for having bad outcomes due to infection? So this is just in general, it applies to any patient with essentially any inborn error of metabolism. So if you have difficulty with swallowing or breathing, if your immune status, essentially your ability to fight off viruses, your infectious contacts, what type of inborn error you may have, your hygiene, whether you're hospitalized or not, and then access or devices. So do you have indwelling lines like catheters, for instance, which can be a source of infection? And for SARS-CoV-2 or COVID-19, this is some of the data from the study that we did to identify what some of those risk factors are during this pandemic in patients with mitochondrial disease. So one of the risk factors, of course, is being in the hospital or healthcare settings, as I mentioned. And during the COVID-19 pandemic, about half of patients still continue to have doctor's visits, which obviously are necessary because they have very serious chronic illnesses. Public contact during COVID-19, 38% of children and 33% of adults have a household member who's an essential worker. So in other words, someone has to leave the household every day and engage with the public. And what about kind of comorbidities? Well, here are some risk factors for severe COVID, which we found in our patients with mitochondrial disease. So 60% of children and 82% of adults reported at least one condition, which is recognized by the CDC for having severe COVID-19, which includes respiratory muscle weakness, immunodeficiency, or asthma. At the end of this survey, we actually asked, what was your greatest concern about COVID-19? And this I'm sure, is shared by other patients and caregivers within the IEM community, is that they're terrified that their child will contract COVID-19, requiring hospitalization, and may experience a metabolic crisis and lose their skills, essentially, and have their lives changed forever. And so therefore, this is kind of a major theme that emerged when we kind of asked an open-ended question about individuals' fears regarding COVID-19. So what is it about viral infection that essentially can be detrimental to kids with mitochondrial disease, as well as other inborn errors of metabolism? Well, metabolic decompensation, right? So here I'm showing you an extreme scenario. This is not one of our patients. This is just a picture that I took from Google. But the reason I put it there is because it really does demonstrate, the level of care that actually may be required during these episodes of metabolic decompensation. So these can be extreme situations, which can be life-threatening. And in mitochondrial disease, it can cause bioenergetic failure, lactic acidosis. The disease can actually get worse. You can have organ failure, such as liver failure. You can have problems with the central nervous system. Patients can develop something called metabolic stroke, and there are complications that occur afterwards, which can be long-lasting. Patients can require extensive ICU care, and these are usually caused by infection. And the treatment in general, there's no magic bullet. It's essentially symptomatic. You're kind of treating the different things which may come up during infection. So another problem is that infection increases energy requirements, and many different patients and caregivers with inborn errors know this, that if you get a fever, you're going to require more calories. So this comes from a study from a long time ago, from 1935, looking at different infections and how your metabolic rate actually increases. So for every 1 degree increase, and this is in the Celsius scale, for fever, the metabolic rate can increase 10% or more. Right. So if you have a fever that goes roughly 2 degrees above, right, you're talking, anywhere between, 20 to 30% increase in your metabolic rate. So therefore you require more calories. And there's a real problem with this because a lot of people don't feel like eating when they're sick. And then also this inability to generate energy is compounding these factors. Infection can also lead to rapid shifts in your energy requirements. So here, this is a study looking at mice, essentially, because obviously we can't infect humans, so we use the mouse as a model. And this mouse was injected with something that simulates a viral infection, and we're looking at body temperature. And you can see here that after you inject this substance, body temperature goes up for a couple of hours after that. And you may say, well, this has to do with, the mice are running around and therefore they're kind of, generating, a lot of body heat that way. Well, actually, this is during a time when they were actually sleeping. So this increase in temperature happens relatively quickly over a couple of hours. It's essentially a mouse fever, and with that comes an increase in your metabolic rate, right? Fever increases metabolic rate. So the point of this slide is to show you is that these changes can happen actually quite rapidly. So the liver is a really important organ in different inborn errors of metabolism and fatty acid oxidation disorders and mitochondrial disease. And hepatic metabolism or liver metabolism actually can change during infection. And the liver is an important organ, right? Because it also helps maintain things like blood sugar and levels of ammonia in the blood and all different sorts of things. So it's a really central metabolic organ. And basically it can change during infection. And one thing that infection does is it leads to metabolic reprogramming. So this is looking at lactate production in the liver, or basically increase of glycolysis, your increase of burning sugar in the liver. And when you inject something that convinces, this is a mouse, by the way, this is the outline of the body of a mouse, and this is the outline of the liver, which is here in pink. And when you inject a substance that simulates a viral infection, the immune system gets activated, the mouse feels miserable, et cetera. What you see is an increase in lactate production, an increase in alanine production, which tells you that burning glucose is working in overtime, and that's reflected here by an increase in the red color. So the color goes from blue to red because you're essentially burning more sugar. And this happens really, really quickly. So much like our changes in temperature, these changes in basically utilizing glucose occurs very quickly. Infection can also damage mitochondria, right? So this is also a similar experiment in mice where you inject the mouse with this agent that simulates a viral infection. And this is looking at mitochondrial function. So in other words, complex 1,3 function, the ability of this oxidative phosphorylation complex to work. And you can see here that when you inject with this substance that simulates a viral infection, the level of activity of complex 1 and complex 3 goes down quite significantly. So in individuals with mitochondrial disease or even fatty acid oxidation disorders who have low mitochondrial function or oxidative phosphorylation to begin with, this can be depressed even further during a viral infection. Okay, so the immune system makes these things called text messages, they make these things called cytokines. I call them text messages, but this is how the cells communicate with each other. And in this picture over here on the left, we're showing these cells communicating with each other. These little tiny white bubbles here are cytokines, and these are the immune cells actually communicating with each other with these really small peptides and proteins, or these text messages. Well, the problem is when different organs in the body get exposed to these cytokines, they can actually have negative effects. So these are actually liver cells from a mouse that we actually put into a dish, and then we dumped a cytokine on it. There are many different types of cytokines. The one that we dumped on it was something called TNF alpha. And this is a cytokine that's really important in inflammation and fighting off infections, et cetera. Well, when you do that and you measure mitochondrial function in these liver cells that have a cytokine dumped on it, this inflammatory cytokine, you can see that the ability of oxidative phosphorylation actually decreases. So the immune system, needs to make these things, these cytokines, to help fight off a viral infection, but they're the bystander effect in organs such as the liver, which may be affected by these cytokines, where it actually decreases the level of oxidative phosphorylation. Another thing that cytokines can do are inhibit fatty acid oxidation. So what I'm showing you here on the left is these are liver cells that are isolated from a mouse. And when we take those liver cells and we put in a virus, this virus is an influenza virus. What you can see is the accumulation of these red dots, right? So what these red dots are fat. There's an inhibition of fatty acid oxidation when you have the addition of an infection like influenza in liver cells. And so, influenza doesn't infect liver cells. It actually activates immune cells that live inside the liver, and they make cytokines. And those cytokines actually then act on the liver cells. And that's exactly what's happening here. And that cytokine that's really important is TNF alpha, which I mentioned before. And when you take the same liver cells and you dump TNF alpha on top of it, right? So in this case, we're not giving the infection, we're just giving the end result of that infection, which is the cytokine, which is TNF alpha, you get these red dots again. So once again, an inhibition of fatty acid oxidation, which is due to the cytokine. Okay, so we've done a lot of studies on this and published a number of studies. What is the response to viral infection in different metabolic pathways? So glycolysis, right, which helps you burn sugar. As I showed you, when you have a viral infection or a simulated viral infection, it increases, so you're going to burn more sugar. In the mitochondria, stuff tends to go down. So oxidative phosphorylation, or the ability to, extract energy from your food, that actually goes down. The urea cycle, which is important for getting rid of ammonia, that goes down, as well as fatty acid oxidation. So we have a model of mitochondrial disease in the liver. And similar to what I showed you before is that when you give that mouse model of mitochondrial disease in the liver an infection, their mitochondria get really big and look terrible, and they get fat. These gray globules here is fat deposition. And I'm showing you the same thing here on the right. It's just with a stain to stain fat. So basically they get fatty liver disease when they get this viral infection. And as I mentioned before also, this is due to a cytokine, which is basically cytotoxic. It is toxic to the liver cells. And you can see here, not only does it work in healthy animals, but the animal that's infected, which is the bottom row here, which is affected by mitochondrial disease, it's much, much worse. They are much more sensitive to this cytokine in terms of its ability to inhibit fatty acid oxidation and cause fatty liver disease. Well, can we block this at all? Well, there's a treatment called etanercept, which is a TNF alpha blocker. So we basically infect the animals and then give them this TNF alpha blocker. And you can see here that the mitochondria here in the lower part of the electron micrograph, the mitochondria go back to normal. They look normal and they don't look distorted like this center picture here. And then the fat disappears. So this red dye here, which is showing fat accumulation in the liver, actually disappears. So this is just showing that the cytokine TNF alpha is toxic to the liver, and if you block it, you can get rid of a lot of these deleterious effects of infection. Okay, so how can we protect ourselves, or how can we protect patients with mitochondrial disease, as well as fatty acid oxidation disorders, against viral infection? So the first thing, of course, is education. With any health condition, education is really, really important. So things like, before the pandemic occurred, the inborn error of metabolism population already knew about social distancing, right? This is something that they were intimately familiar with regards to staying away from individuals who were having infections or had signs of infection. We also learned about masks, reduction of risk exposure. So in other words, not going into settings where there are a lot of individuals, hand washing, and then also recognition of comorbidities. So in other words, not only do individuals with inborn errors have their inborn errors, but they have other things too. They may have things like asthma, which, put them at an increased risk. The second thing is adherence to the childhood vaccination schedule. So this is important for protecting children against common illnesses of childhood. Situations to discuss with your provider with regards to the vaccination schedule. So if there are live vaccines, basically with documented immunodeficiency. So if you're an individual who's concerned about live vaccines, such as the measles, mumps, rubella vaccine, and you have documented immunodeficiency, you should discuss that with your immunologist. Those with a history of anaphylaxis, so severe allergic reactions. This is something also you should discuss with your provider with regards to vaccines. And then a history of adverse reactions. So in other words, if in the past, individuals who've had difficulty with vaccines, with getting fever and that fever causing difficulty in their disease, this is also another situation that you should discuss with your metabolism provider. We use antibiotics a lot in different inborn errors of metabolism to help with infections or even for prophylaxis to help prevent infections, as well as antiviral drugs. So during flu season, there are some providers who will actually prescribe Tamiflu because Tamiflu has to be taken early during influenza infection for it to be effective. So some individuals actually have that in the home. Some patients with mitochondrial disease may actually require immunoglobulin therapy, right? So immunoglobulin therapy are basically those antibodies that I was talking about, that I was showing you. The ones I was showing you were naturally produced ones, but what immunoglobulin is it's basically antibodies that are pooled from anywhere between 10 to 100 different healthy individuals, and it's given as a drug. And that is given for a number of different immunologic conditions, including immunodeficiency. So there's an intravenous form, so where you get it in the vein. There's a subcutaneous form. And as I said, it's produced from human plasma, from anywhere between 10 to 100 individuals. And it's used to treat immunodeficiency. It's kind of a little bit of a black box in terms of how it exactly works, because there are many other effects of immunoglobulin therapy, which we just don't understand yet. So what's interesting is that when we looked in our patient population of mitochondrial disease, there were a number of individuals who were on IVIG or subcutaneous immunoglobulin therapy. So we looked by our VIRSCAN platform to see what were the number of different viruses that they potentially could be protected against by looking at those antibodies, by pulling down those antibodies. And this is the figure that I showed you before, right? Where control and patients with mitochondrial disease, it's roughly about 15 viral things that they've been exposed to over their lifetime. But when we look at kids with mitochondrial disease who are being given immunoglobulin therapy, right? So this is a mixture of their own antibodies and antibodies from other individuals. They basically can potentially be protected against roughly 20 different types of infections. We don't know how effective it may be, but what we do know anecdotally from individuals who are on immunoglobulin therapy is that they do experience less infections. Okay, so in summary, unfortunately, viral infection is inevitable. I showed you that patients with mitochondrial disease have roughly 15, at the age of 6, have been exposed to roughly 15 different viruses, which is the same as individuals in our control population, which kind of tells you, a lot of measures that patients will use to stay away and try and prevent infection, infection's inevitable. It's going to happen sooner or later. So we need to understand it, and we need to find new ways to be able to protect patients. Viral infection can be detrimental to patients with mitochondrial disease, as well as many other different types of inborn errors of metabolism. And then predictors of negative outcomes due to infection, we just don't know what they are. We don't know what individuals are going to have trouble with infection and what individuals are not going to have trouble with infection. So that's why we're working on it. And it's likely multifactorial. Many different factors are involved. What we can do in the meantime is we can offer anticipatory guidance, vaccination, and treatment of infections to try and maintain health in patients with mitochondrial disease. So I'd like to thank you. This is the team who has done a lot of work basically to learn about viral infections in patients with mitochondrial disease. And I'd be happy to take any questions. Thank you.
Well, thank you, Dr. McGuire, as always, very informational. And I truly admire all the work that you've done to help understand how our kids and their immune systems work, or in some cases fail. We have a lot of questions here, so I will go through them. Could Dr. McGuire comment on how infections could cause mito dysfunction versus having mito disease and then getting an infection?
Yeah, that's a great question. So mitochondrial dysfunction during infection is not unique to mitochondrial disease, and it's not unique to inborn errors of metabolism. It actually occurs in many different situations. So if you've ever heard of like sepsis, right? During sepsis, that's overwhelming infection in the body, and it happens. It's a real big problem, and patients are admitted to the intensive care unit. And these are individuals who don't have inborn errors of metabolism. Because of the immune reaction that's going on, right, and it's really robust and overwhelming because of the overwhelming infection, the cytokines that are made during that can actually inhibit mitochondrial metabolism in various organs. So people will get organ dysfunction or failure, or multi-system organ dysfunction or failure, which I'm sure you've heard of. So yes, it is a situation that is not unique to inborn errors of metabolism. It does occur in individuals who don't have it, but in that particular case, it's more because of an overwhelming infection and an overwhelming immune response. The difference with inborn errors of metabolism is that threshold is much lower. So in other words, they don't necessarily require, for the immune system and the damaging effects of the immune, we need the immune system. We definitely need it to get rid of the infection. But as I said, there's this bystander effect, where the tissues that are affected by inborn errors of metabolism may be more sensitive to these, different aspects of the immune system, which can cause the cells to become dysfunctional or die.
Okay. Do you recommend a daily dose of vitamin D for children under 12?
Right. So most of us are vitamin D deficient in general, myself included, right? It's because, I don't spend much time in the sun. The sun is a major source. The sources of vitamin D are, sun, and then also food, but the bioavailability in food can be tricky. So in general, we are lacking vitamin D. While I can't give specific advice on anyone's, specific case, these are things that you discuss with your pediatrician. So they should look and see, how well-rounded is their diet? Are there any things that may raise risk of or raise concern about potentially being vitamin D deficient? That's what I would recommend, rather than giving a general recommendation.
Yeah, I know those of us north of the Mason-Dixon line are just permanently deficient.
Right. And and with my complexion, right, I can't spend a lot of time in the sun. So, right. And a lot of other people may share that issue as well.
Yep. How likely can a virus cause rhabdo, even if there are no other symptoms of the virus or illness?
Yeah, that's a great question. I mean, the answer is we really don't know, right? So in other words, are there viruses where it's subclinical infection, right? So in other words, you don't experience any, other, issues, but it causes rhabdo? That's a great question. Unfortunately, I don't know the answer to that. And I think it's an interesting question, and certainly an interesting area of study, but I don't think there's really any information out there right now to that I could share with you.
All right. Are there things we can do to stop a viral process once it's started?
Yeah, so you can't necessarily stop it. You can help support, the body's ability to fight it, right? So in other words, it's basically those sick day care regimens that individuals will be recommended, and a lot of times it has to do with increasing calories, right? As they pointed out, when you have a fever, your basal metabolic rate increases significantly, right? It can increase up to 30% or more, depending on how high the temperature goes. So you want to support that. Nutrition is certainly a really important aspect of that. So getting those calories in and being able to, support that, really, I would say, is probably one of the most important aspects of helping to deal with infection in terms of things that you can do.
Is staying hydrated equally as important as the intake of the calories?
Yes, absolutely. So staying hydrated as well is an important part of that. Usually that comes with the calories in a sense, right? Because usually the calories are somewhat driven by, taking stuff that you can drink essentially. The idea is that there's a deficit, right? And a deficit in energy, and you have to try and make up for that. And usually that does happen through hydration essentially.
Yeah. That's the tricky one when they're sick, to get the nutrition in there. You're right. They don't want to eat. They don't want to drink
because they don't feel good. They don't want to take it in. And that's, a common reason for a lot of ER visits, right? Because it's difficult sometimes to keep up with that requirement.
Right. Is there anything we can do to give the B and T cells a boost to help them kill the viruses quickly?
Yeah, I mean, really, it's there's no magic bullet, really. The T and B cells work on their own time scale in terms of how they react to infection and their ability to get rid of infection. But once again, I think this comes back to nutrition as well, where it's working with your dietitians to not only manage, your disease, but also make sure that your health and your kind of nutritional health in general is actually good as well. And so some nutritionists will do kind of like a yearly kind of more extensive evaluation, right? To look at things like iron and micronutrients and all other stuff. These are things that actually are important for the immune system to function, things like selenium and copper and all these other things. So once again, I think, especially in terms of fatty acid oxidation disorders, it comes back to nutrition and realizing that you have to restrict certain things out of your diet, obviously, as part of that disorder. But there also should be a goal of looking at overall nutritional health and trying to, improve that or maintain that as much as possible.
All right. And I have one last question here. Why do you feel that some doctors are reluctant to prescribe or even entertain immunoglobulin? I can't even say it. You know what I'm saying?
Immunoglobulin therapy. Yeah. Well, yeah. It's no small undertaking, right? It really isn't. It's So what are some of the issues? There are essentially two ways you can get it, right? You can get it by IV, which is like once a month. So there's problems with access and potentially getting infections with IVs and stuff like that. And then there's subcutaneous, right? So basically it's under the skin, and then eventually it kind of makes its way into the blood, and that's actually more protective in the sense that it lasts longer and you don't have kind of these spike and dips of immunoglobulin. But once again, that's putting something under the skin, and it's, it comes with its own set of complications and infection risks. Okay, that's number one. Number two is some patients don't tolerate it, right? So you can have an allergic reaction to immunoglobulin therapy. And some patients actually need to be pre-medicated before actually receiving immunoglobulin therapy, where they get things like Benadryl and Tylenol and stuff like that, which, considering fatty acid oxidation disorders, sometimes, some individuals are, that's not good for that, like things like Tylenol and stuff with liver function. That may not be a good idea. Right. So there are complications that come with it. And then, of course, what was my last point? Prescribing it, right? It's not easy to prescribe. So in other words, it requires an infusion or subcutaneous infusion, which requires a nurse, right? Which is usually done. It can be done in a home healthcare setting. It can be done in a hospital. But then the last thing is you have to qualify for it, right? So for insurance to pay for this, you have to qualify for immunoglobulin therapy. And, therefore they have to have demonstrated by an immunologist a need for that immunoglobulin therapy. So otherwise it will not get paid for. And one infusion, I think, is about $10,000. So
Right.
it's there are a lot of factors around it, but those are some of the main ones that I think are important in highlighting.
Yeah, it is not an easy undertaking, that is for sure. Well, we are out of time. I have numerous questions that we didn't get to. I will try to get those answered, and maybe we can ask those during the panel discussion later this morning. Does that work for you?
That sounds good.